Where's yeah, it's fine now.
Okay, sorry for technical interruptions. So we're talking about biological development. In other words, question is, let's say, if I take hair or skin or stomach and do gene analysis, genome analysis will be the same genome, right? Despite the fact that this is different, right? Our hair is very different. So the question, how the same set of genes, so the same DNA can produce very different morphologically different cells. That's how the different tissues appear from initially identical, genetically identical cells. So the biological development is a problem which still captivates people in different areas, from biologists to chemists, physicists, computer scientists, mathematicians. So for example, for physicists, this is a problem of so-called large-scale emerging phenomena. For computer science people, mathematicians, this is the problem how information is passed, right? So it's the distribution of information, because the organism, when it's developed, is essentially information written in DNA. You have to pass it so they can build properly the organism. And for engineers, an example of very robust system. We talked about in the first lectures about robustness, right? So these are very robust systems. Okay, good. So let's talk about another important issue which I didn't talk before: universality. Let me try to explain. So there are many different airplanes in the world, right? There are military, civil, small, large, whatever, a lot of them. All of them look very different, but they have one thing in common. There is one mechanism how they fly, jet engine mechanism, right? So there is always a question comes, biologists and asking the question, are phenomena in biology the same way? They might look very different, but do they always use the same mechanism or not? So this is a question of universality. So if you ask any biologist, you can ask your biological friends, they will tell you no. Every new biological phenomenon is completely different. If you ask physicists or chemists who actually doing biological, they will tell you yes, there is universality. Okay? I don't know what is the right answer, but I want you to be aware of this universality issue. And fortunately, this biological development is probably universal. Probably we can say with a large degree of confidence it's universal. And let me show you the picture, which is almost 200 years old. There was a scientist in Russia. His name was Karl Ernst von Baer. So he was a German from a Baltic country, from Estonia. And so these are embryos of different organisms, and you have to look from the top to down. Okay, so this is from the fish, salamander, turtles, chicken, hawk, whatever, right? You can see there's clearly similarity, clearly universality, something, right? All these embryos are developing in a similar way. This, by the way, is actually a very important picture. This is the picture that when Charles Darwin looked at it, was one of the things that stimulated him to develop evolution theory. Yeah, so that was Karl Ernst von Baer. This is this person, but a Russian scientist who made significant contribution to biology. He's essentially father of embryology, science about embryonic development. So as I told you, that this picture was something, one of 2 or 3 things that stimulated Charles Darwin to develop evolution theory. the most surprising thing that this person was the strongest opponent to Charles Darwin evolution theory. Surprising. So this is a monument to Karl Ernst Baer in Tartu. Tartu is a city in Estonia, which in older time was called Derg, because the old university, very nice university. So there is a very interesting tradition. You know what is a Walpurgis night? No, have you Wagner? Do you like Richard Wagner, composer? You know, so there is in a German. Okay, I know Indian mythology is extremely complicated. German mythology, also complicated, and there's a special night. The special night is between April 30 and May 1st, called Walpurgis night. This is the night when all the witches are flying. Anyway, so the students in this university have a tradition. Tartu University, they washing Karl Ernst von Baer with champagne. See, that's a picture, a real picture. So this monument is really shiny because every year on May 1st, they wash this person with champagne. Sometimes with a beer, but mostly with champagne. This is champagne. Good. Morphogens. Okay, let's come back to science. Morphogens. One of the central concepts in biological development is called morphogens. It comes from a Greek word, two Greek words, morphos, change, gene development. That was actually proposed by Alan Turing in 1953. So this is a compound, molecules which are responsible for cellular fate. You can call them dose-dependent gene regulators. So the idea that goes back to Alan Turing was, imagine these are different embryo cells, but all of them identical. Then there is this embryo cells have to produce these chemicals, morphogens, they're diffusing, and then there is a non-uniform potential. And then the idea is if you're this concentration region, you turn on gene A, but not other. If you this concentration region, you turn on gene B, but not, and so on. So in other words, this explains why there are different tissues. So our genome, you can think about the genome as a set of bulbs. In stem cells, they're originally turned off. And then comes this development process, morphogens, and they turn on specific labs. If you red one, you will have it here. The blue one, you will get skin, whatever. That's a general idea of development, how it works. That's why different morphologically different cells have the same genome. Okay, so Alan Turing, you're in the department of mathematics. He's the most famous person, right? What he did for computer science? No, before. What is the most important contribution to computer science and mathematics? Huh? Algorithms. He is the person who said, essentially started algorithms. But he was a very talented person. He did a lot of things here. He made a lot of contribution to theoretical biology, especially to pattern formation and concept of morphogens. But he's one of the most, not the most important, but the most famous achievement was cracking the code of this machine, Enigma. Do you know what is this machine? Excellent. You're a very educated person. So during the World War II, Germans had a machine which was doing codes, so they could send it to their agents, right? And they thought that nobody could break it. He was able to break it. So the British were able to read codes. And the machine was called Enigma. This is real Enigma, how it looked like. And that was his most famous achievement, but not the most important from my point of view. So he was really genius. Good. So essentially, starting from Alan Turing and other people, there is this concept of positional information comes in biology. The idea is, as I already mentioned, so this morphogens create non-uniform profile, and this non-uniformity is a way of passing information. This non-uniformity is a way of passing information, and threshold comes into play. Those exactly the same thresholds that I mentioned. So because if your concentration above this, there is one gene activated, if this is another gene. This is called French flag model. That's because the flags are French flag. And by the way, this is experimental. You can see. So this is the embryo of fruit fly, and you can see from after some experiments that some of genes are activated and in position dependent. In this case, this morphogen was called BCD or bicoid protein and a different concentration of bicoid protein stimulated different genes. Again, thresholds here. There's a threshold. Okay. Yeah. So this idea of positional entropy was introduced by a very famous person called Lewis Wolter. I don't know if in India he wrote a lot of science popular books. I don't know if they're Indian, not that I have seen them even in Russia. He was very excellent writer, popular books about science. Yeah, so that's Lewis Wolter. Lewis Wolter, and he was born in South Africa, Jewish person, then essentially worked most of his life in the United Kingdom. If you have a chance, strongly recommend you look at scientific books or give it to your kids. It's very nicely written, very pleasant. He's actually also credited with a famous quote, so you can read it here. So it's not the most important event in human life is not a birth or marriage or death, but gastrulation. Gastrulation is the process when you are developing different tissues. It's called gastrulation. Okay? So those people who are concerned with the marriage, don't worry. The most important things already happened to you. Gastrulation. Okay. Anyway.
Professor, you were saying that what I understood, you said that morphogens decide which genes are turned on and which are off.
They don't decide, but they stimulate a cascade of events that eventually activate different genes. And there is a, so there is this, it's a proof, experimental proof. You see, so here is bicoid, and you can see bicoid has different concentration. I can show a better picture, but you can see that bicoid is more at this end than this end, right? And then eventually you turn, this is one gene, this is another, this is the third one. It's indirectly.
Did you also mention something about diffusion of the morphogens? And
that will come. It will come. It will come. Yes. So this is an example of complex patterning, which is a result of all this complexity. There is information is transferred through non-uniform concentration. Yes. So this is a better picture which comes from my friend, who is a professor at Princeton. He's my friend. So he again, it's a fruit fly. You understand, you cannot work on human embryos, right? You will be killed because it's unethical, right? But fortunately, you can work, so most people who do science work on embryos of fruit flies or worms. That's the typical things. Fortunately, it's the same event in embryos of humans or chicken here. So he did beautiful measurements. So in this case, this bicoid is a green one. Okay, you can see there is more of this bicoid here than over there. The important thing is that there's a non-uniform concentration. And this non-uniform concentration is a way to pass the information. And there is another protein called nuclearization, and there is another NATK. Anyway, so that comes to your question. So what people think about this? So one of the most important events, how you can establish this morphogen gradient. So you need to have this, by morphogen gradient, I mean non-uniform concentration of the signaling molecules. And the simplest idea, which goes back to Turing, it's something which in this field called synthesis diffusion degradation. This is nothing else as reaction diffusion for some people who already talked with some people, right? It's a reaction diffusion. But I also want to notice, don't you think it's the same, almost the same what I told you this morning about catalysis? Remember? Was a diffusion plus death. It's the same thing, right? So the bottom line is, using the same mathematics, you can solve very different problems. Okay? So I write this equation here as a continuous time equation, differential, right? And before I was writing the master equation in a discrete language, but it's the same thing. I want you to understand. Okay, good. So essentially the diffusion, degradation, and this is the concentration of morphogens. And if you try to solve the equation, you can get it easily. There will be this decay. Again, it's a stochastic processes, right? Look, the stochastic processes governing these events. And that's what's happening in more microscopically. So there's a set of cells. One of them started randomly produce this morphogen. And then they outside of cells, then they diffuse, and they go to other cells receptors. They are removed. They remove more. As a result, you will have decreasing the concentrations. And then depending on how many of them enter this, you can activate one set of biochemical pathway here or another one, which leads to activation of different genes. Okay, so what's not working? Good. Okay, so what are the problems with this field? So people were able to measure many of these things quite well, and there was a problem. So one of the most studied systems is called this bicoid proteins and fruit flies. So when they measure diffusion coefficient, they could measure diffusion coefficient, and they know the total length. So let's come back to this picture so you will understand. So people know the total length of this embryo. Okay, they know that bicoid is produced here and eventually has to diffuse. This distance close to 400 micron. So the problem comes is that 375 microns people know, they know diffusion coefficients. Then you can estimate time it takes for the molecules from one end to reach another one, right? You understand, right? And you do the square distance of diffusion coefficient, that's the estimate. But simultaneously, people were able to measure these things, and it's only 90 minutes. So it's like somebody on a bike moves as fast as somebody on a BMW, which is strange, right? So that's a problem. That's a problem, and this is called paradox. So there is slow diffusion and quite fast formation of morphogen gradient. So this non-uniform profiles form quite fast. People thought about this, proposed several mechanisms, but none of them agrees with experimental observation. Good. So one more piece of information comes, which is given by this very famous person, Francis Crick. So what Crick is famous for? Francis Crick? Good. DNA, correct. So Francis Crick was concerned with the following question. Timing. It takes some time before morphogen gradients can be created. And it is important to have essentially a stationary profile because you need to pass your information robustly, right? You need to have robust information. But how long does it take? It was not clear. Okay, so Francis Crick, very famous person, Nobel Prize for discovery DNA double structure. By the way, this is a fig photo that I made myself in a bar in Cambridge, where apparently Crick and came and with his collaborator Watson Crick, they came and apparently announced it in a bar. Very British way to announce what they did in a bar. So there is a sign here. That's what I made. Is it here? Anyway, he was actually a very interesting person. He was very smart with a lot of things, but he had a lot of strange things. He was extremely critical of religion, especially Christian religions. I don't know why. He also had this view. He was promoting something that people call positive eugenics. Let me explain it in a simple terms. So if husband and wife both of us smart people, they should have kids. If they're not, they should not. Anyway, this is. But he had Nobel Prize. He could do whatever he wanted at this moment. Anyway, so let's go back to the problem of establishment of the gradient. So essentially, the problem comes to the fact how long does it take to establish a stationary state in a specific place. So this problem of morphogen gradients, you start everything here, but then eventually your system, how long does it take to establish a stationary state at some location away from the source? And in 2010, very smart work came out, and people were able to establish it. They introduced so-called relaxation function, and they had this time. They were able. You can calculate time to establish a stationary state in a specific location. This is a different time scale. We didn't talk about this. In all the things that I told you, I actually assumed that you immediately go to stationary state. But that's a non-trivial problem. How long does it take to establish a stationary state? And okay, so applying to this reaction diffusion model or standard diffusion degradation, synthesis diffusion degradation, that's the answer they got it. So this is the time. So imagine this is the distance from the source. This is the time to establish a stationary state distance x from the source, and so k is the degradation rate, x is the position down to the specific length. So doesn't look familiar. Remember when I talked about catalysis? It was the same length scale came out. Remember? Square root of diffusion over this. Again, I want to emphasize different mathematics. I mean, different problems, the same mathematics. So there is a time scale, so there is a length scale here. Good, everything is fine. This problem partially explains the problem of fast formation of gradients, but there is something surprising here. The linear scaling. So molecules have no bias. They can equally go here or here. If there is no bias, the diffusion should be square. Size x squared should be proportional to time, not linear. You expected quadratic scaling. You expected power law 2 here, but you get only 1, which means that there's a kind of a force or potential, and it's not clear where it comes from. The molecules has no bias. It's morphogen molecule. Yeah, so this is my naive explanations of, we talked about the above, but let me try to revise it again. So unbiased diffusion, imagine there is a cat which can go left and right, right? So the distance that cat travels in time t travels scales as r squared. That's because you do a lot of redundancy. You do like this, you do like this. So as a result, if you measure the time, the distance you travel will be r squared over proportional t. But if there is some bias, sausage. Okay, so probably I better draw a dog because dog has better. But anyway, let's get who likes sausage. Then it will go in this direction. That will be bias diffusion. In this case, distance will be proportional to time because you mostly go in one direction. Okay, so I'm trying to explain what is the problem. The problem was it looks like experiment shows the morphogen molecules are not biased. But when I try to do the calculations or following these things, it looks like they're biased. So these are the problems. So the measured diffusion constant are too small. The linear scaling, not clear where it comes from. What is the mechanism of this effective degradation? And we explain this using again stochastic model. We can propose a stochastic model, which again is very similar to what was the chemical catalysis I did in the morning. That was the reason for me to give chemical first in the morning and then do very similar, the same mathematics, but different phenomena. There is a model which is exactly birth death with diffusion. So you assume that the signal molecule produced at the beginning. So here the lattice is semi-infinite, in contrast to what I had in the morning. Here the lattice is semi-infinite, start from 0 goes to infinity. But otherwise, you're producing this molecule, they can diffuse and then can be removed, degraded. So it's a reaction diffusion of birth death stochastic process. You can calculate it again. For this model, you can calculate time it takes to establish a stationary state in location M. And it's again linear scaling. There's explicit formulas. And if you want, you can do simpler formulas in some limiting cases. In any cases, there is a linear dependence. And before going, there is something else interesting. Okay, so this is the time to establish a stationary state in location N. And you might ask the question, okay, how this time depends on K, on this degradation rate. And the interesting observation here is that in contrast to naive expectation, if you increase the degradation rate, you actually can reach stationary state faster. So if you increase degradation rate, you can reach stationary state faster. This is strange. Let me give you non-scientific explanation. Was it on TV game here in India? Who wants to be a millionaire, right? Okay. Globalization, everywhere is the same thing. So if you have a large degradation rate, then your stationary profile has a low concentration, very low. Then it's very easy to reach from 0 to low. It's easy, right? If you need in one day get $100, it's easy, right? I mean, I don't know, 1,000 rupees. You can get from friends, they will give you 1,000 rupees, easy, right? But if you need to collect, let's say, 1 million rupees, it's not easy, right? It's exactly the story. So to go from 0 to something small, you can do it faster. That's exponential. Okay. Good. Okay, so let's come back to this. So this relaxation times are times to establish a stationary state. So I'm trying to understand why there is this linear scaling, right? I said experiments show on the microscopic level molecules of morphogen has no preference. It can go equally, probably left or right, whatever. But when I put all the formulas in experimental data, it seems like there is bias. Let me see, how can I think about this relaxation time? I can think about this relaxation time in the following way. This time, we can write it as a two terms. So the first term is this first arrival time, mean first passage time. This is related to the question that you already asked. This is the first passage time, and then so the molecule should come first to this location, then some time for adjustment, and that will be the relaxation time. Okay, and you think about it, so the further away this location, the more important will be this first passage time. So you can do anyway with argument that this relaxation time can be well approximated by mean first passage time if you're far away from the origin. And it's convenient because we can calculate mean first passage times for this problem. So the mean first passage time to start, if you start here, how long it will take for you to reach the location n. And what we did it, and it's again linear, which is surprising. It's a mean first passage time, unbiased diffusion. It's supposed to be quadratic. How it can be linear? And this slide shows that this mean first passage time is a good approximation. And what I plot here, exact analytical calculation for relaxation time versus mean first passage time. And if I go further away from the origin, the ratio goes to 1. So mean first passage time is a very good approximation to yeah. So important things. Mean first passage time is a good approximation to estimate how long does it take to reach a station. Okay, good. So this linear scaling is unexpected, but I was essentially lying to you all the time. The system is not unbiased diffusion because there is a degradation. Degradation does something to the system. Degradation behaves like a potential. It reduces the residence time at each site, and it creates effective concentration gradient. Effective concentration gradient is identical to the force or to potential. Let's try to explain it a little bit more. So imagine that you start at time 0. At time 0, you're producing your morphogens mostly here. As time proceeds, you will have like a kind of a step. Why step? Because molecules which are a little bit further away will be some of them will be already killed, removed. Okay? And if you continue this way at your station, it will be a lot of steps, but you don't see the small steps. So you are removing, by degradation removes particle, and the further away you remove more. As a result, you have effective. So each molecule individually has no bias to go, right? But if there is a 10 molecules here and nothing here, they will most probably will go there, right? So that the degradation creates an effective potential. Let me give politically incorrect Texas explanation in other ways. So imagine that, okay, so imagine that I have a gun and I shoot through this door every second, right? And people are running. Only those who really run fast will survive. You understand, right? Or if you slow, you will be killed. So it's exactly here. This degradation, like a shooting, only those molecules which move faster enough, they will survive. Because if the longer you stay at given site, the more probability you will be removed. So this is another way of looking at acceleration. Degradation works like effective acceleration, like effective potential. And you can actually even calculate this potential analytically in this case, and that's what we did it. Through, if you know the stationary profile, which you know for this problem, logarithm of the stationary profile times the effective potential, and you can do the calculation. This actually means that there is a constant effective force at every location which tries to move particle in one direction. Again, the source is that your system is semi-infinite, and that there is a, if you're moving, the longer you're in a system, the more probability you will be removed. That creates effective potential one direction. Okay. And that's explanation for linear scaling, right? So remember, in the field with the force, you move faster. It's a driven diffusion versus undriven diffusion, using the language of Tasset. Tasset is driven, right? But the symmetric one will be undriven. So degradation creates this effective driven condition. Now, you can actually expand this idea further away, and there are some interesting things. So there are some systems where people arguing that the degradation is not linear but powerful. So essentially, they're arguing that equation that describes the system, it's again reaction diffusion, but in a slightly different way. There's power law here. There's so-called self-enhanced Liebman degradation in a simple molecular way. So the morphogen comes to cell and can only be degraded if another morphogen comes, for example, and some other scenarios. Okay.
Is this the power?
Yeah, well, it's M number. I consider the case so far n equal 1, but it could be 2, 3, 4, whatever. Okay? So this becomes much complicated nonlinear problem.
Non-linear.
Nonlinear problem, but instead of solving this problem, I can do the same trick as I told you, right? I can look at degradation as effective potential, and I can, because I can, all I need to know is the stationary profile, it will be 0. I can get something like this, and there will be some interesting predictions. So the predictions from these simple calculations are the following ones. So if I have m, this exponent, 0 or 1, then the degradation is very strong potential, and you have a driven system. But if m is larger than 1, it's still unbiased diffusion. But what this potential does, it changes effective diffusion coefficient. It changes the effective diffusion coefficient is faster. Okay. This comes from very simple arguments, and then there was a paper written by mathematicians who actually got the same result through complex analysis. But I got it immediately from very simple arguments. Okay, and this is example of this potential. This is effective potential of the functional location. If it's a linear one, you have very strong potential, but those non-linear are relatively weak. That's why you have different behavior. Okay, so essentially, I'd like to emphasize again, we had a stochastic mechanism, right? Very similar to what I introduced in the morning for cooperativity. Same mathematics, almost same mathematics. I can explain mean observations, and I can explain how stochastically you can produce this profile, and what is the consequence of this. And well, we argue that the relaxation times essentially are mostly determined by first passage times, and this degradation effectively works with the potential, and we come out with a possible mechanism of acceleration. It's like a potential, so degradation works as a potential. That's a possible mechanism of acceleration. Again, importantly, stochastic processes are creating complex dynamics in the system. Okay, so the nature didn't care about the nature doesn't care that if you remove or kill this morphogen, right? The nature wanted to do things to do faster, and to do faster you do the degradation. There was problem. You understand all of these things, you are wasting the energy, right? But probably that you need to do the things fast. But that was the system. Okay, so this is it. I didn't want to again to overwhelm you. So this is a different story with essentially the same mathematics. Okay, same mathematics. It's a reaction diffusion model, stochastic model, burst death, and you can get, in my opinion, some better understanding. Okay. Any questions, please? I hope you didn't feel asleep. Please.
So, as far as the morphogen is concerned, it is the production of the morphogen is localized. Is that correct?
Yeah. So people believe that. Yeah. Okay. So it's a good question. So people believe that. Yeah. So some people believe that. Okay. No, maybe even before. Yeah. So people believe that there are several embryo cells, and then at some time something happened in one of them randomly, and it started to produce, and others do not, and that's enough to establish. That's something what they believe. So the stories are more complicated. Biology is a complicated thing, but that randomly you started somewhere, and then it diffuses. So it diffuses and removes, creating non-uniform profile.
Second thing is in in some of those chemical kinetics you had talked about earlier, there were some similarities in the equations at the macro and the micro scale. And I guess you can go from the micro to macro in some scaling limit. I guess here what I was wondering is the microscopic model you had with the lattice, where you have the production Q and then you're hopping.
Yes.
Yes, this one. Is this kind of a parallel of that?
It's more general. As you understand, discrete phenomenon, discrete is always more general. I can always go from discrete to continuous by making the lattice size smaller and smaller. That will be the limit. Continuous is always approximation. People always forget about it. So I don't know about here, but in Russia we thought that it's easier to solve differential equation instead of difference equation, right? But strictly speaking, different equation is more general. You can get differential one from a different, but not another way around.
And one more thing here, the birth is only happening at 0. Is that
That's correct. So this is it's mimicking what I told you just before, that one of the cells started randomly to produce it, and that's what it is. Right. So and again, so important things here. This length scale appears, right? This is average distance before morphogen gradient molecule will disappear, and it's exactly the same distance that I had in the morning as communication length in catalytic, right?
This is the solution to the second order differential equation. And
This one,
time derivative.
no, this is actually even easier. A little bit,
Yeah.
yeah. So this is you calculate this relaxation function.
Yeah.
So this is a stationary constellation. This is instantaneous constellation. You don't even need to calculate. You can do essentially Laplace transformation, and then you can get this time. And then from this time, this exact formula comes. Right. This exact formula comes. I also derive the formula for discrete version. So this was for continuum version, right? For discrete version, this is the formula that I have. If you want to go to the limit, diffusion much larger than degradation, that will be continuum limit, and you get the same result. Right. But there's a
Suppose we have a system who has a steady state, okay? To reach that particular steady state, we started with some initial condition, and then with certain MFPT first particle time, we reach to the steady state. If we change the initial condition, so the transient state, again it will reach to same steady
it
state.
will have to
So
reach,
that-
unless there is some special system by stable, which happens, but not here. That you will get to the same stationary state.
So my question is, the MFPT will come same or it
Yeah.
is going to come?
It is. It must be the same. Yeah.
It is the average of all the first passage times.
Yeah.
First
It's average over all
You are
possible
performing-
trajectories.
yes, You are performing multiple simulations in the platform and take the average.
That's correct.
That's why it is called.
So we were the
The transient analysis we don't do much of. In Tesla
transient to get up to
There is a transient for it. Time part.
question from
It's not chemistry. I hope it's easier for many people, right? But yeah, please.
Sorry to pester you. Are there any textbook treatments that come to mind that might cover some of these things? Quantitative biology here.
No, I can send you my review paper where I try to explain it, but otherwise, not to the best of my knowledge, but probably it is somewhere. So there is a good textbook which is so it's a Conde Phillips, Yanne Conde Phillips. It's cell physical biology that has a little bit of it, but it's a little bit. Okay, so again, I want to emphasize my point was to present chemical story intentionally in the morning, and to show this exactly the same mathematics, right? With a different biological afternoon. Again, everything is stochastic, and you still can produce things. You can calculate. You can explain the formation of the gradient. You can explain time. Okay.
thank you.
It's on a particle. I'm